The overall goal of the following experiment is to fabricate sodium nanosensors for in vitro and in vivo applications. This is achieved by preparing an optode mixture containing the sensor components for the nanosensors. As a second step, surfactant is sonicated in aqueous solution to evenly distribute it, which will allow nanosensor formation when the optode is added. Next, the optode mixture is added to the aqueous surfactant mixture under sonication to form the nanosensors in solution. This video demonstrates that the sensor response is tunable, the sensors can be micro-injected into cardiac myocytes, and that the sensors can also be injected subcutaneously into mice.
The main advantage of this technique over existing methods, like fluorescent indicator dyes, is that we can tune the sensors to a dynamic range that matches physiological environment, measure a wider range of analytes, and perform ratiometric measurements for quantitative analysis.
Generally, individuals new to this method can struggle because once the sensors are made, it can seem daunting to introduce them into a biological environment.
Stocks of dry sodium ionophore X, dry sodium TFPB, and chromoionophore 3 need to be prepared in advance of making the optode. According to the written protocol, in a fume hood, combine all the materials into a 1.5 ml glass vial with a Teflon-coated screw cap. Gently vortex the vial until all the PVC has dissolved. The solution should appear red. Begin making the nanosensors by adding 100 microliters of 10 mg per milliliter PEG lipid into a 4-dram glass vial. Next, add 4 milliliters of the desired aqueous solution, such as 10 millimolar HEPs. Sink a sonicating tip 7 mm into the vial using a laboratory jack and apply a low sonication power for 30 seconds. In a 0.6 milliliter tube, combine 50 microliters of the optode material and 50 microliters of dichloromethane using the pipette to ensure even mixing. Next, begin a 3-minute sonication. As the sonication begins, add 100 microliters of the optode mixture by immersing the pipette tip and dispensing the complete volume in one quick, even injection. The solution will turn blue if the pH rises above 7 and if there is no sodium. To remove the residual THF and dichloromethane from the solution, move the sonicating tip to a depth of 2 mm. As the remaining 3 minutes elapse, the solution will become opaque. Once sonication is complete, pull up the nanosensor solution into a 5 ml syringe. Attach a filter and dispense the nanosensor solution into a glass vial with a screw top cap. Begin by making 10 solutions with progressively increasing sodium concentration, against which the sensitivity of the nanosensor can be measured. Combine 0 molar and 1 molar sodium stock solutions in 50 ml conical tubes. Now, in an optical bottom 96 well plate, add 100 microliters of each concentration of sodium to 3 wells in a column. Then, add 100 microliters of freshly prepared nanosensors to each well. Load the plate into a microplate fluorometer and enter the excitation, emission, and cutoff wavelengths that are ideal for the chromoionophore being used. The choice of measured wavelengths depends on the application. These wavelengths provide for intracellular fast dynamics, measurements, or noise reduction. To obtain alpha values, divide the ratio of intensity at 570 nanometers by the intensity at 670 nanometers or 680 nanometers. Plot the alpha values against the log of the sodium concentration, setting the log of 0 sodium concentration to -1. Now, fit a sigmoidal curve to the response, and from the curve, calculate the sodium concentration where alpha value is 0.5. This represents the sodium concentration at which the sensor responds optimally. Using this information, adjust the ratio of sodium ionophore X, sodium TFPB, and chromoionophore 3 in the optode to create nanosensors with the optimal sodium concentration. For example, here are response curves of 5 different nanosensor sets representing different optode formulations varying only in the amount of sodium ionophore 10. For intracellular imaging, replace the cell media with a clear media like Tyrode solution and transfer the cells to an imaging chamber. Now, backfill a pulled glass pipette with nanosensor solution made in 5 mg per milliliter glucose in water. Then, mount the pipette to a micromanipulator and connect it to a pressure-controlled injection system. Lower the pipette on top of the cell and into the cytoplasm. Sometimes it is necessary to tap the manipulator holder to pierce the membrane. Inject a few hundred picoliters of nanosensor solution and quickly withdraw the pipette. Anesthetize a CD1 nude mouse with isoflurane using an anesthesia chamber. Once anesthetized, place mouse into disinfected imaging chamber. Next, fill a sterile 31-gauge insulin syringe with 10 microliters of nanosensors with the appropriate KD in 10 millimolar PBS. Make sure to remove all air bubbles. Using forceps, grasp a small fold of skin along the back of the mouse at the desired injection site. While grasping the skin, insert the syringe into the skin with the bevel facing up and the needle parallel to the skin. Now, pull back on the syringe plunger to ensure the needle is not inserted into a blood vessel. To minimize the back pressure, gently move the needle right and left to create a pocket within the skin, then inject the sensors and gently remove the syringe. Gently apply pressure to the injection site and wipe away any solution that may have leaked out of the injection site. Typically, 6 evenly spaced injections are made along the back. If performing on different mice, change the needle to minimize disease transmission and cross-contamination. After the injections, acquire bright field and fluorescent images using filter sets that closely match the excitation emission spectrum of the nanosensors. This also minimizes the autofluorescence of the skin. These neonatal cardiac myocytes were cultured on a cover glass and injected with sodium nanosensors. This fluorescent image is taken at a 639 nanometer excitation wavelength. This is a bright field image of the same cells. And this is an overlay of the two. There is some sensor clustering that occurs, but the cells have normal morphology. Sensors have diffused throughout the cytosol, and there's no nuclear loading. This overlay of a bright field and fluorescent image shows 9 different subcutaneous injections of sodium nanosensors into the subcutaneous space of a nude mouse.
Once mastered, fabricating nanosensors can be done in minutes, if performed properly, and applied to any research to measure reversible, real-time analyte flux.
After watching this video, you should have a good understanding of how to load nanosensors into the intracellular or subcutaneous space for quantitative measurements in a biological environment.